My name again is Ben Adam. I'm a renal pathologist at the University of Alberta in Edmonton, Canada. And on behalf of the Renal Pathology Society, it is my honor to welcome you to the third session in our latest webinar series, which is focused on focal segmental glomerulosclerosis and APOL1 nephropathies. As I mentioned at our previous sessions in the series, our understanding of these diseases has rapidly expanded in recent years, and these new insights have significant implications for renal pathologists, nephrologists, and patients with kidney disease. So the intent of this initiative is to provide a cutting-edge update on these important topics to our colleagues and partners around the world. And I would like to acknowledge that this series is supported by an educational grant from Vertex Pharmaceuticals, so thank you very much to them for their support. So just a few quick housekeeping items before we get started. First, we have arranged for live interpretation of the session into both Spanish and Mandarin Chinese, so please be sure to select your preferred language within Zoom. Second, we should have time for a short question and answer session following the presentation, so please enter your questions into the Q&A section and we will do our best to address as many as we can. Finally, you will be asked to complete a brief survey at the end of the webinar, so please take a minute or two to fill this out because this feedback is very important for planning future sessions. So without further ado, it is my great pleasure to introduce today's speaker, Dr. Matt Sampson, who will be discussing the genetics of podocytopathies, emerging technologies, and novel insights. So Dr. Sampson received his BSc in Cell and Molecular Biology at Duke University and his MD at the University of Virginia. He spent the next 6 years at Children's Hospital of Philadelphia and the University of Pennsylvania, where he completed his residency in pediatrics, his fellowship in pediatric nephrology, and a master's degree in epidemiology and human genetics. After 8 years on faculty at the University of Michigan, where he established his kidneyomics lab, he moved to Boston Children's Hospital and Harvard Medical School in 2019, where he is a nephrologist, associate professor of pediatrics, and holds the Warren E. Grupe Chair in Pediatric Nephrology. He's also an associate member of the Broad Institute, where he's a member of the Kidney Disease Initiative. He is the co-chairman of the Genetics and Genomics Working Group of the Nephrotic Syndrome Study Network, or Neptune, and the Kidney Disease Working Group of the ClinGen Consortium. His research is primarily focused on mapping the genomic determinants of nephrotic syndrome through integrated system genomics and multi-omic methods, and to discover the molecular, mechanistic, and clinical consequences of them when they are found. He's also committed to improving genomic literacy among pediatric nephrologists and other clinicians and maximizing the utility of genomic testing in the clinic. So thank you very much, Dr. Sampson, for being with us here today, and I will hand it over to you.
Thank you. Thanks very much for the invitation. A couple of comments. One, I really enjoy speaking to the RPS. It was really the renal pathologist that really got me ultimately into pediatric nephrology, as a kidney is not something that I could feel, palpate, hear, listen to, or observe clinically. It was really making those connections between the patients I saw at the bedside and their kidneys on light immunofluorescence and electron microscopy that really brought me into the fold. So I really appreciate y'all. I also just want to note that the last time I spoke to y'all was on February 29th, Leap Day 2020, which was in California. It was the last trip I made before the COVID-19. That day, they had just announced that someone had been infected with COVID-19 who didn't have any connection to China. And I think we knew that the world was changing. I don't know if it was that much, but it's good to be back. I'm glad that we're all safe. This is my talk. Here's my lab website, Twitter handle. You guys can feel free to tweet anything here. All right. So the objective here is really to contextualize the known genetic causes and contributors to nephrotic syndrome within the framework of genetic architecture. I really hope that when you leave this talk today, you have an understanding about the real underlying genetic architecture of this condition. And what do I mean by that? So what I mean by genetic architecture is the overall composition of the variant influencing given trait. Here it's nephrotic syndrome. We're talking about the number of genetic variants associated with the condition, their effect size, their allele frequency in the population, rare to common, the concept that there may be interactions between all the variants contributing to a disease, and the possible interactions between variants and environmental factors. And while we'll focus on nephrotic syndrome podocytopathies today, recognize that this concept of genetic architecture is generalizable to whatever condition of interest to you. And as I go through the genetic architecture of nephrotic syndrome, I'll try to highlight some of the emerging technologies that empower this work and share some newer concepts and insights that are resulting from these efforts. So, our group's drive to take a kidneyomics approach to nephrotic syndrome is really driven by the massive frustration of the imprecision of the current classification nephrotic syndrome. We as clinicians characterize our patients based on renal biopsy findings of a histologic description. So if there are minimal changes, we call a patient having minimal change disease. Or if there's focal segmental lesions of glomerular sclerosis, we say that the disease FSGS. Or if there are different immune complexes, we give it that name. These are histologic descriptions, which you don't need me to teach you about or tell you more about, but this does not give us an idea of the molecular underpinnings of the condition. In the same way, response to immunosuppression, it's really, where the horse is out of the barn by the time we start making these diagnoses. Patients, we give steroids and they're steroid sensitive or resistant, and then they could be primarily resistant or secondarily resistant. We can give steroids over a course of months to years and classify patients based on their relapse frequency. They can jump between steroid sensitivity to steroid resistance. And we have really no great idea of why this is happening. The treatments we give, we have no certainty of whether they're going to work or not, but we know for sure that the non-specific therapies we're going to give are going to have side effects. So it's this lack of precision, and my training and coming up during medical training during the publication of the Human Genome Project and working in a PEDS hemonc lab where they were doing this molecular medicine bench to bedside and back in the early 2000s, that really led me to be obsessed with using genomics to dissect the molecular anatomy of nephrotic syndrome. This was heavily influenced by folks doing monogenic disease research as well. So that being said, our group has chosen over the last decade to go after nephrotic syndrome from a genetics perspective. And there's two major ways, in my opinion, to get after genetic causes or contributors to kidney disease. One is through the study of families, where there are multiply affected individuals over generations who harbor the disease, and then doing genetic mapping to discover causal genes. Or a more recent advent is using populations to discover genetic variants that contribute to disease. A family-based approach is treating nephrotic syndrome as a Mendelian disease, and we're using techniques such as linkage or positional cloning. Historically, more recently, doing whole exome sequencing and taking a more genome scale approach with a little bit less emphasis on the linkage positional cloning that dominated early genomic discovery. When we're using populations to discover genetic drivers of nephrotic syndrome, we're more treating nephrotic syndrome as a complex disease in which there are variants that are present in the population that increase risk but are not sufficient to cause disease. When we do this genome-wide, looking for common variants, this is called the Genomewide association study. And when we do look at it for rare variants, it's called burden testing or rare variant association studies. So what's the value of unraveling the genetic architecture of nephrotic syndrome? Well, I think you all inherently know this, but I'll just articulate it. It's really to discover known and novel NS-associated genetic variants and trying to understand how problems with these genes or these regulatory regions cause or contribute to kidney disease in humans. Once we can map these genetic contributors or causes to nephrotic syndrome, we can do a couple of things. We can more immediately do clinical associations or phenotypic consequences. If we have sufficient patients who have been phenotyped, we can look at clinical associations such as associations with demographic characteristics, age of onset, for instance, baseline characteristics, eGFR, urine protein to creatinine ratio, baseline, longitudinal outcomes like decline of eGFR over time, response to immunotherapy, recurrence after transplantation. And we can also stratify our patients for clinical trials, such as doing things like trying different immunotherapies for patients who do not have a Mendelian form of disease, or more recently, as was reported out yesterday in a phase 2 pilot study, a study by the drug company Vertex, who's doing APOL1 directed therapy for patients who have a high-risk APOL1 allele. So it's the stratification of patients for clinical trials, which I think is going to be increasingly powerful and almost a necessity, in my opinion, for moving trials forward. And I want to note that you don't even need to know the molecular mechanisms that are driving this genotype to disease association to enroll people or to classify patients and have it be helpful. Longer term, of course, we don't want to just map the genetic variants that are associated with nephrotic syndrome. We also want to understand the mechanisms that link it. And this is the concept of mechanisms to medicines. And by doing this, we can define biology experimentally and through integration with kidney tissue-derived human data sets. That's a lot of the work that we do that's been pioneered by folks in Neptune and in the KPMP project and others. We want to identify targets for therapeutic intervention. You might notice that I continue to talk about, genetic variants that cause or contribute to disease. And that, again, is getting at genetic architecture and recognizing that there's a range of genetic variants that range from, which I'll talk about soon, variants that are almost deterministic, that if you have the genetic variant, you have the disease, so that's a cause. Or there are more common variants that are contributory, which require either a second genetic hit or some environmental stimuli or interaction to cause disease. A novel insight, I think, over the decades, excuse me, that's not even true. A novel insight over probably the past 5 or 10 years, as we've sequenced more and more healthy individuals in the population who do not have kidney disease, is what we're recognizing is that members of the people in the population also harbor variants that we would predict to be pathogenic or disease-causing. So the idea of incomplete penetrance or variable penetrance is an emerging insight. And it's leading us to think that less and less are there genetic variants that are 100% causal, and it's more likely that things are, in many instances, have a range of penetrance. Some are very, very penetrant, but a lot of things are not absolute. Happy to talk about that more later. So here's how I want you to conceptualize the genetic architecture of podocytopathies. You could also do this for kidney stone disease, tubulopathy, cystic kidney diseases, etc. So please just think about genetic variants on the axis of allele frequency, how often this genetic variant is seen in the population, and if that genetic variant is present, its effect size on the phenotype of interest. So this ranges from alleles that are very rare or absent in the population that, when present, have a huge impact, including being deterministic, and those are rare alleles or novel alleles causing Mendelian disease, going all the way to common alleles in the population that are present in many of us in this audience today, which are not sufficient to cause disease, but increase the risk in the presence of other unknown factors. We can map our family-based and population-based studies onto this figure, and recognize that family-based studies are great at discovering these Mendelian alleles, and population-based studies are empower our discovery of common variants with small effects, common variants with larger effects, as well as variants that are lower frequency in the population, maybe 1 to 0.5%, that nonetheless have a moderate impact or effect size. And I'm going to carry this figure forward throughout the rest of the talk to map genetic contribution to nephrotic syndrome on it. So the nomenclature, historically people talk about mutations. I think that's a colloquialism, it's pretty imprecise. I think what I prefer, most of us in the field would prefer, rather than calling things mutations, are pathogenic variants or rare pathogenic variants. So pathogenic meaning that it causes a problem, it causes a disease, and rare lets us know that that it is not common in the population. Risk alleles is another colloquialism. Some people call them risk SNPs, risk alleles, but I think it's better to call them common alleles in the population, low frequency alleles in the population that have a small or moderate or large effect size. So if you and your trainees can think about characterizing using this kind of terminology, it's a lot more descriptive than saying someone has a mutation or a risk SNP or a polymorphism. So let's talk about these rare alleles causing Mendelian disease and nephrotic syndrome. And really, The Mendelian forms of nephrotic syndrome are those that are associated with steroid-resistant nephrotic syndrome, or FSGS. And we make this distinction because many kids with SRNS are not, or they're not always biopsied, but the clinical phenotype is more associated with Mendelian disease. And identifying these rare alleles and families over the decades in our field collectively has really allowed us to identify. This is a study of the coding regions of the genome, the exons, and we could figure out directly what protein is broken with these disease-causing pathogenic variants, and allows us to understand biology. And in doing so and thinking about nephrotic syndrome as a rare Mendelian disease, SRNS, we've mapped, collectively have mapped mutations or genes that harbor pathogenic variants to, initially the slit diaphragm with nephron and podocin, the actin cytoskeleton, actin binding proteins, more recently CoQ10 biosynthesis, and nucleoporins. And there are now about 60 single gene causes of steroid-resistant nephrotic syndrome, or FSGS, that have been discovered. Well, understanding this and finding patients who have these genetic variants, these pathogenic variants, are important. There are clinical implications. These patients are much less likely to respond to immunosuppression, and they're also less likely to recur after transplant. The question is, these are all, it's very important that these genes have been discovered to help us understand biology and ultimately to go towards mechanisms and medicines. But how about the clinical import for those, those clear genotype-phenotype associations that I just mentioned with immunosuppression and therapy? So thankfully, people have done work on this around the world. And when looking at steroid-resistant nephrotic syndrome, one form of podocytopathy, the most comprehensive study by Sadowski et al., I think in 2014, which was from Dr. Friedhelm Hildebrandt's lab, shows that the prevalence of Mendelian forms of steroid-resistant nephrotic syndrome really range from about 45% in Middle Eastern countries to about 13 or 14% in the US. And this is a direct correlation with the degree of consanguinity in the population. So regions of the world in which people who are related tend to have children, the prevalence of Mendelian forms of nephrotic syndrome is much higher. Or in populations that are less likely to reproduce with people outside their population. In Boston, a follow-up study of SRNS, 11%, the UK and Turkey, 23 to 26%. We did a study, the Neptune study, which is a more unselected population of patients, it was 6%. And you can see in Brazil, which is another large admixed population with not a lot of consanguine endogamy, it's about 8%. When looking at these prevalence estimates, it's also another insight is you really, when reading these, you have to understand where these numbers are coming from. We recognize that the percent of patients with Mendelian forms of nephrotic syndrome are much higher in congenital nephrotic syndrome, less than 1, and much lower as we go through adolescence. So some of these studies have included patients with congenital nephrotic syndrome. So really an insight is when counseling your patients, reading out reports, the chance of having a Mendelian disease is really influenced by the chance that they are from a consanguineous union, as well as the age of onset of the disease. An important aspect is, this is steroid-resistant nephrotic syndrome, which is the minority of cases of nephrotic syndrome in the world. When we think about immunosuppressant-sensitive nephrotic syndrome, whether it's steroid-sensitive, steroid-dependent, frequently relapsing, monogenic forms of this disease are far, far less than 1%. There are very few genes, if any, which harbor pathogenic variants for immunosuppressant-sensitive NS. So that's just not a form of genetic architecture for this condition. Now, when we move from rare alleles with high effects, in nephrology and podocytopathies, we really have an exceptional situation here because there are a number of common variants that exist in the population that, when present, nonetheless have a high effect size. And this is the story of membranous nephropathy and APOL1 associated FSGS. So a genome-wide association study of membranous nephropathy, which was done in 2011, looked across the genome, and each one of these dots is a common variant in the population. And the y-axis is showing the odds of having membranous nephropathy in the presence of this variant when doing case control study. Repeatedly replicated a signal at the HLA locus in chromosome 6, and then at this locus at chromosome 2 in the region of PLA2R1. And it showed that individuals with this HLA-DQA1 variant had a 4.3 times increased odds of developing membranous nephropathy. The PLA2R1 variant, when present, had a 2.3 times increased odds. And when an individual in the population was homozygous for both these risk alleles, so 2 bad risk alleles in this variant, 2 risk alleles in this variant, they had an 89 times increased odds of developing disease, which is quasi-Mendelian. It's important to recognize that these variants are common in the population. So people are walking around with this in the population and have an increased odds of disease. And subsequent work, now in 2020, work by Shi et al. Coming out of the group of Christoph Kierluck at Colombia, with collaboration from the UK, identified in now 3,800 cases more genetic loci associated membranous. We have HLA, PLA2R1 again, and now we have a signal NF-kappa beta and IRF4. Again, these are great mapping studies, and if we do mapping, then we can then take these genetic loci and link them to mechanisms, and we could also link them to clinical correlates and clinical stratification. And as you all, or most of you, are aware, the work on PLA2R and PLA2R staining and serum levels have really revolutionized the therapy and treatment of membranous nephropathy. So what we look forward to doing in the future as we unravel this genetic architecture of membranous nephropathy is to take these signals forward, and this is a GWAS, so these are not rare coding variants. These are often regulatory variants, non-coding variants. What we need to do once we find these peaks, and these gene names are really the closest neighbor to these variants, we have to implicate the exact genes that are targeted or impacted by these risk alleles, the regulatory variants that are impacting these genes expressions, and the cell type that they are involved in. So that's another insight, that the genetic architecture of podocytopathies are certainly impacting kidney cells and podocytes within of themselves, like PLA2R1, but some of these genetic factors that underlie podocytopathies are also having their causal effects by influencing immune and circulating cells. So we need to be able to dissect this as well. So now we'll move to our other story of the APOL1. So APOL1 was initially discovered through work by Cow et al. And Winkler et al., where they said, we recognize that Black individuals have a higher incidence of FSGS in the population. Therefore, we believe that any disease-causing variant for FSGS in Black individuals will become in parts of the chromosome that are enriched for African ancestry. And in doing this admixture linkage study, which is what I just described, they found there was a locus on chromosome 22 among Black patients with FSGS versus Black individuals without FSGS, where those with FSGS had a much higher percentage of African ancestry at chromosome 22. And MyH9 was originally linked to this region, and then follow-up studies fine mapped this region from the work from Martin Pollack's group and David Friedman and Carl Skorecki's group. They identified that it was APOL1 that had these common risk variants that arose about 10,000 years ago in sub-Saharan Africa that were associated with 15 to 20 times increased risk of FSGS, 30 times increased risk of HIV-associated nephropathy, with a collapsing phenotype. So it was really, again, the genetic architecture recognizing that there were ancestry-specific or enrichment of disease in certain ancestry groups that allowed people to do clever mapping to discover these variants. The fact that these variants, 13% of all Black individuals in the country have a high-risk genotype, and the odds ratio of having disease is about 10 to 15%, means that APOL1 high-risk variants are very common cause or contributor to disease in FSGS patients in the US. So if all these individuals here are all the Black individuals with FSGS we see in our clinics, in both adults and children in multiple studies, we recognize that about 70% of our Black FSGS patients in the population have a high-risk genotype. So we were really invested in knowing the clinical correlates of this, but we're also invested in understanding the mechanisms that link the APOL1 high-risk genotype, which are exonic to disease. So getting at some of the emerging technologies, which isn't all that emerging, it's using transcript. We can use, what we can try to do is link the glomerular transcriptome to the disease phenotype, to link the to try to understand the mechanisms underlying APOL1 associated kidney disease. So some work that we've done in the Neptune study. This is a visual abstract, which we just had this paper accepted at KI, so it should be coming out pretty soon, led by Michelle McNulty in our group. But really, this is a study of the Neptune network. What we did is with our renal biopsy specimens, we looked at gene expression in the glomeruli of 30 Black individuals in Neptune. All 30 individuals had FSGS, and all 30 individuals were Black. What's going on? Okay, and there were 14 individuals were low risk APOL1 genotype, and 16 individuals were high risk APOL1 genotype. And we had the hypothesis that there would be different glomerular transcriptomic signatures between these groups of individuals that could illuminate some of the biology underlying the disease. So we looked at differential expression between the two groups. We looked at shared and inverse correlation of genes with APOL1 in the high risk and low risk state. We looked at differential co-expression between the two groups. And this is all online on bioRxiv, but I'll focus on the most easy to interpret sub-analysis we did, which was the differential expression. So we simply asked, out of almost 16,000 genes expressed in the glomerulus, do individuals with FSGS and the high-risk genotype have a differential expression of any genes as compared to the low-risk genotype? This is a volcano plot showing each dot is a gene. On the right side are genes that have higher expression in high-risk APOL1 FSGS, higher expression in low-risk FSGS, and this is Genomewide significant. What we found is 9 significantly differentially expressed genes, and these were the 9. And actually, most of them were well, that's not true. There were some that were decreased expression in high risk, like CCL18, and then those that were higher expressed in the high risk included genes like STC1, CCN3. What we noticed here is that the sets of genes that were upregulated in high risk APOL1 were more mapping, doing functional analysis of the literature and bioinformatic tools. They're cytoprotective genes that regulate mitochondrial function, as well as those that impact calcium regulation and are potential upstream triggers. So we decided to focus on the most significantly increased gene expression in the high-risk glomeruli, stannocalcin-1, which we know plays a role in mitochondrial and calcium-related processes. And previous work has been shown that it's upregulated in cases of AKI, and seemed to be the focus of its study has been a lot on tubular expression. But we worked with our local friendly neighborhood collaborator in pathology, Astrid Wines at the Brigham. And she collaborated with us and looked at STC1 staining in a normal kidney. I'm going to walk you through this light micro. No, I'm just kidding. I'm not going to walk you pathologists through this. I'll just say that in normal kidney, STC1 expression was not really appreciable. This is a qualitative approach based on visualization. In collapsing FSGS with no APOL1 risk alleles, STC1 did not have much higher expression than in a normal situation. But in collapsing FSGS with 2 APOL1 high-risk alleles, STC1 had substantially increased expression that looked diffusely detected in endothelium. This is preliminary work that needs to be followed up and further examined, but is giving us a hint that as we try to link genetic changes to disease mechanisms, that going through the glomerular transcriptome may be a way to get after it. I just want to highlight that everything I just discussed with regards to APOL1 can be found on the APOL1 portal that we created with Jason Flanick here at Boston Children's Hospital and the Broad Institute. The full paper is there for download, and you can also download all of our RNA-seq data, all the covariates. You can interactively search results, and you can explore correlations, which gets at another major thrust of my lab's research mission, which is to have as widespread and broad data sharing as possible to parallelize discovery so that as many folks as interested can really try to help discover the kiniomic basis of nephrotic syndrome with us. So another genetic architecture of nephrotic syndrome are low-frequency variants with an intermediate effect. So these are variants that are present maybe at 1% of the population, 2%, maybe 0.5%, that are not, again, sufficient to cause disease, but could have an intermediate effect. So there's one study that was done by you et al. This was led by Andre Shaw when he was at WashU, who's now at Genentech. And what he did was an FSGS rare variant association study among European individuals. This is just a plot showing that they did a good job of matching European cases of FSGS and European controls, and simply used burden testing to look to see if there's specific genes that had an excess enrichment of pathogenic variants in cases versus controls. Came up with a list of about 10 genes. And in 2016, this was more supportive evidence that COL4A4 variants could be associated with FSGS alone and not necessarily Alport syndrome or any obvious basement membrane disorder. CAT2B, which was subsequently discovered as a Mendelian nephrotic syndrome gene, but also interestingly in these European patients, self-described white, genetically global ancestry white, found APOL1 as risk alleles. This is the G1 risk allele as being increased in white FSGS patients. And what we know now as we continue to discuss and think about race and what that means, the social construct of race and structural racism as it impacts kidney health, what we recognize here again is that APOL1 risk alleles are not necessarily restricted to those who self-identify as Black. We recognize that individuals who are of Hispanic descent, especially Caribbean descent, have a small but substantial prevalence of the high-risk APOL1 genotype, but even individuals who might describe themselves as white or would have the possibility of having high-risk APOL1. So that's another novel insight that I think we need to keep in mind, that just because someone doesn't self-report as Black doesn't mean that they cannot have APOL1 disease. But again, what this is implying to us is that there's another group of genes that can have variants that in the right circumstances can cause FSGS, again, in the context of a second hit, whether it's environmental or genetic. Now let's talk about steroid-sensitive nephrotic syndrome. So steroid-sensitive nephrotic syndrome is really, the genetic architecture has been described now over the last 4 or 5 years as defined by common variants in the population that have moderate effect sizes. So this is 5 GWAS studies that I just want you to kind of squint your eyes and you can see these peaks that look very similar parts of the genome. And if you were my students and we were live, I'd quiz you and I'd say, where do you think this is at? And well, I wouldn't end the sentence in a preposition. I'd say, where do you think these are? But this is in the HLA region. And these are studies that are in from South Asia, led by Rashid Gabatagisan from Duke and the Midwest Pediatric Nephrology Consortium, Hanna Debiek from the NEPHROPATHIES Consortium, and Pierre Ronco and Francesco Emma in Italy and Europe, and our group at Neptune. This is by Robert Clayton, Detlef Bockenauer, and others in the UK, and this is Kazumoto Ijima and Dr. Katsushi Tokunaga out of Japan. And what we're seeing is in children with SSNS around the world, of all ancestries, the lead variants are risk variants are always in the HLA region and MHC. And we're starting to discover other variants outside of the MHC region, which are starting to help us understand a little bit more the biology of this disease, again, leading to mechanisms and clinical growths. This paper, which we were fortunate to be a part of, which was published in KI in 2020, did a GWAS of Japanese patients with steroid-sensitive nephrotic syndrome, replicated in Korean children with SSNS, and then followed up in European, Black, North African individuals, and South Asian. Found, again, the HLA region was associated with SSNS, but common variants in this tumor necrosis factor gene, as well as nephron and QRL2 were associated with disease. Again, these are non-coding variants. These or their coding is synonymous. So we have to understand what this is telling us. Of course, nephron, which is one of the fundamental genes of the slit diaphragm and is one of the first, the first reported cause of congenital nephrotic syndrome and also involved in SRNS, was incredibly intriguing to us because we've never really thought of nephron as an SSNS gene. And we looked more deeply into this study, and we found that all these risk alleles seem to map. This is a gene-dense region, so we can't be sure that it's nephron. And we know QRL2 also has impact on, or part of glomerular biology. But given that all the top SNPs, most of them are nephron introns and nephron exons, including 2 synonymous variants, we felt more certain about going after nephron to understand how common variants can impact the gene's function and contribute to SSNS. And there's actually another insight that supports this idea is this concept of, well, this study here, this is a genome-wide association study of diabetic kidney disease. They looked genome-wide in patients with type 1 diabetes and controls, and they found a bunch of genetic loci associated with DKD. And one of the most significant loci was a low-frequency exonic variant that was missense in COL4A3, which is a fundamental, basement membrane gene associated with Alport syndrome. So this was replicated, and this showed that while rare, novel pathogenic variants in COL4A3 can cause Alport syndrome, more common variants in this gene can be associated with a more complex trait like diabetic kidney disease. So we saw this. Uromodulin is another gene that, rare mutations or pathogenic variants can cause autosomal dominant tubular interstitial kidney disease. More common variants are associated with CKD and hypertension. So there's this paradigm that maybe, severe mutations can cause rare Mendelian diseases. More common variants can contribute to more complex conditions. So we really, again, emerging technologies. We took advantage of RNA-seq data, whole genome sequencing data to try to, again, understand why we're seeing this signal in nephron. And we first asked, are these nephron coding and non-coding variants, are they changing the expression of nephron? Are they EQTLs? That's what EQTL is. Or do these nephron variants cause alternative splicing, so spliced QTLs? And this was not apparent from our data out of the Neptune study. We had, again, through the prescient planning of design of Neptune, in which we had extra renal biopsy cores and RNA-seq generated, we were able to see that the 4 individuals in Neptune with this nephron risk haplotype did not have different expression as compared to those without the risk haplotype, which was the easiest answer and perhaps the most common way that GWAS, non-coding GWAS alleles are associated by changing gene expression, but we didn't see that. We also did not see any examples of exon skipping, which is illustrated here, or new exons being spliced in as a result of this risk haplotype. So it was not as easy as these nephron risk alleles are changing nephron expression or causing a new isoform. We then asked about allele-specific expression, which is not a common genetic mechanism that's implicated in the genetics of nephrotic syndrome at this point, but which we're kind of going after these days. So allele-specific expression can only be done if you have gene expression information. And it's the idea that if this is, you have 2 chromosomes, one from mom and one from dad. Golly, why does it keep switching like that? Interesting, it has a mind of its own. That ordinarily you might have a 50-50 transcription of nephron from the maternal chromosome and paternal chromosome, but there may be an imbalance of mRNA transcription, and that might be pathogenic for any number of reasons. And we can measure this using phased whole genome sequencing and RNA-seq BAM files, and we can see if there's deviation from the norm. And if in a normal situation, we can say the nephron expressed is equal between mom and dad's chromosome versus imbalanced. So we asked if there was allele-specific expression associated with nephron. Again, this is all published. So this is a graph on the y-axis. If it's down here, that means there's no allele-specific expression. If it's up here, it means all reads are coming from one chromosome. And what we saw is samples without the risk haplotype did not seem to have much evidence of allele-specific expression, whereas the 4 individuals with the risk haplotype did have allele-specific expression. This was very significant, or it was significant. And just a reminder, we did not need genome-wide levels of significance. This was just a single hypothesis test. And when we looked more closely at these 4 individuals, the risk haplotype is in dark blue and the reference haplotype is in light blue. And we can see for all 4 individuals that individuals with nephrotic syndrome who had the risk haplotype had less nephron being expressed from that chromosome. So what we observe is less mRNA resulting from the NPHS1 risk haplotype. And we don't have any idea right this second why this is a problem. We are engaged in work right now to try to understand. We can hypothesize maybe that this chromosome is somehow malfunctioning and resulting in inability to transcribe, and nephron is so important that the reference allele chromosome is overworking to produce more. That could be causing cellular stress. There may be things with, these are synonymous variants, maybe things with translational pausing. It's all hand waving based on review of the literature for other conditions, but the bottom line is, for us, this is a high value target to continue to discover, to continue to work on, to see if maybe we can identify mechanisms underlying SSNS and maybe get away from just bombing all our patients with steroids to try to treat their disease non-specifically. So I just want to briefly talk about future directions. I think we've kind of gone through the genetics of podocytopathies from different genetic architectures. I think this is what we need to do to continue to unravel the genetic architecture of nephrotic syndrome as a world community. What's the role of structural variation? So we know that deletions, duplications, transversions, translocations are a prevalent contributor to other kidney diseases like congenital anomalies and other diseases outside the kidney. Are duplications and deletions a big part of nephrotic syndrome genetic architecture? Is this COL4A3 story and the nephron story paradigmatic or one-offs? So will we continue as we build our sample sizes and cohorts? Are we going to find, again, that regulatory variants or hypomorphic alleles or less severe variants in known Mendelian disease genes, what integral podocyte proteins, are they going to be contributing to complex diseases in ways that we don't understand? To continue to unravel the genetic architecture, we just need to get more patients, particularly from underrepresented populations. The global South has been really underrepresented in genomic discovery work. Taking more patients and those from underrepresented populations will allow us to discover new single gene causative diseases, discover more loci from genome-wide association studies. It will empower rare variant association discoveries. We might be able to discover modifiers in which there may be a rare mutation in a known pathogenic variant in a known gene, but maybe the presence of a modifier can alter the clinical severity of the condition. We also need to facilitate data sharing through meta-analysis, that will allow meta-analyses that will bring in additional families to replicate discoveries. We wrote a position piece or an opinion piece about using and producing publicly available genomic data. I think if you look to other diseases around the world and other traits, whether it's the CKD Gen consortium with EGFR or type 2 diabetes, schizophrenia, there are other fields that are doing a real great job of from a position of PIs, authors, funding agencies, journals, of really empowering and data sharing and enforcing data sharing. And I think we in nephrotic syndrome and glomerular diseases are getting better and better. We're building cohorts, we're working together well, but we need to continue to figure out ways that we can do this. That's a win-win for everyone, especially, and it will ultimately be a win-win for the patients. And getting more patients and getting more kidney tissue, which I showed the value of, is one of the reasons I moved my lab from University of Michigan to Boston Children's, because I had the opportunity here to build a cohort that we're calling the Big Kid Study. It's the biobank to investigate the genomic basis of kidney disorders. It took 2 years to build in the context of COVID, but now it's open, and it's based I base this on the philosophy that every patient should be able to be involved in nephrotic syndrome research, whether they're at a big center or at a community hospital, from both a patient and clinician perspective. The Big Kids study, anyone with nephrotic syndrome is eligible, from children to adults, steroid sensitive, dependent, resistant. You can go on our lab's website, the samsonlab.org, or you could look into Big Kids here, and you can directly contact us, and we'd be happy to enroll patients and ultimately involve people in the data analysis, paper writing and discovery. And I think in doing this is a rare disease. This is not hypertension in adults where I could just walk over to the Brigham or Mass General or the BI or BMC and get like 1,000 patients in a year. We really need to get patients from, Bali to Bosnia to Bahrain to the US and Canada, all my beat countries I ran out of. We need to get as many patients as possible into these studies. We also collect kidney tissue and do work from there. So I'm hoping that this moving forward the next 5 years, especially in pediatrics, which has been understudied, we can build these cohorts to do more discovery to get after that. So my final comments on genetic architecture, this is my last slide, is precision phenotyping is just as important as genotyping. I think genetic discovery or genetics will never supplant the importance and the role of kidney biopsies. It may allow us to shape who we biopsy and when, but especially from a research perspective, having very clear histologic diagnoses is really empowering the kind of discoveries and podocytopathies we're doing right now. We also need to, moving forward, we need to maximize the accuracy of the classifications of genes and variants as disease-related. So there's a lot in the literature that's very valid about genes that harbor variants that are disease-causing, but there are unfortunately more papers or more entries into databases that are questionable or are frankly incorrect that we need to really clean up. A recent paper commentary led by Danny Gale out of the UK and Detlef Bockenauer, myself, and Andrew Mallet of Australia, talked about these diagnoses of uncertain significance. And the major thrust of the ClinGen Consortium, our kidney disease working group, is to make sure that when we identify variants that we have a more certainty whether they're pathogenic or not. The kidney disease working group of the ClinGen Consortium is wide open for anyone who wants to take part in variant adjudication. You can feel free to contact me or look us up online to be part of it. We have a number of renal pathologists involved in this work as well. And finally, I think we need to be aware of attributing causality or disease modification from combinations of alleles across nephrotic syndrome genes. So now that we can do panels of genes and whole exome sequencing, we're going to start to see this buildup of variants across different genes that our desire to tell just so stories are going to make us want to say, oh, this patient has a single variant in PLCE1 and one in LAMP2 and one in PHS2. All those three heterozygous variants and recessive genes together, they must be causing the disease. Or a lot of times we're seeing APOL1 variants in combination with COL4A variants. We're saying, oh, well, APOL1 is a modifier of COL4A or COL4A or modifiers of APOL1. It's all really possible that this accumulation of variants that alter proteins across genes could very well be a form of nephrotic syndrome genetic architecture, but this has not been formally tested or formally proven at this point, with some rare exceptions. We know that people in the population who don't have disease have these variants as well. So again, we need to increase sample size. We need to bring more kidney omics investigators into this world and increase rigor to model this, to decide that ultimately whether or not this is a form of disease. But all this is doable. I think the strides over the last 15 years, from me starting as a very eager resident or fellow in nephrology, peering through the microscope with Dr. Russo at CHOP, now to being here at Boston Children's with a collection of great pathologists and nephrologists in a worldwide community. We're data sharing, we're working together, we're making discoveries, and it's an exciting time. So with that, I'd like to thank you. This is my crew here. Feel free to be in touch if you'd like to discuss any of this more, and if there's time, I would love to be able to take some questions. Thank you.
All right, thanks so much, Matt. That was a fantastic talk. I think that the approach that you and your colleagues are taking is really the model and represents the future of not only nephrology but medicine in general. And yeah, it's really impressive and important work that you guys are doing.
Thanks.
And yeah, I mean, it's a complicated topic, but as usual, you did a great job of making it practical and easily digestible. So thanks for that. We do have a few questions that have come in, a few kudos as well in the chat. So Parmjeet Randhawa has posed a couple of questions. He's already unmuted, so I wonder if, Parmjeet, did you want to ask the questions yourself, or do you want me to ask them on your behalf? Yeah, Parmjeet, you should be able to unmute yourself and speak.
Hey, Ben, you can go ahead and just read the questions. Yeah, that's
okay,
fine.
yeah, I'll just moderate as usual then.
Yeah.
Okay, so the first question is, I think you might have addressed this in your last couple of slides, but just to confirm, where can clinicians working in the community send samples for genetic studies when indicated?
Yeah, so there are a variety of companies or hospital systems that can do testing ranging from single gene testing to panel testing to whole exome sequencing. It's just going to depend on what hospital system you're in, what state, what country you can do them. So that's for clinical genetic testing. For research genetic testing, you can send samples to my group, samsonlab.org. You can email me for that. These days, we're really no longer in the situation where we can return research genetic testing to clinicians for clinical management. So that's less of a thing these days. In terms of insurance coverage, that's the ongoing question. It's a major thrust of my lab. One of the postdocs in our group, Jen Fishbein, is working on trying to understand how we can systematize and operationalize insurance coverage. Right now, it's a bunch of people who have ways of understanding the local situation to try to get insurance coverages, but it's always a challenge. My hope is one day we can systematize this to allow this to be done. I think we need to make an economic justification why doing genetic testing is important and saves money to allow this to happen.
Great, thanks, Matt. That was the follow-up question was asking for a comment on insurance coverage. So yeah, thanks for addressing that as well.
Yeah.
So the next question is, congenital nephrotic syndrome has a significant recurrence rate after kidney transplantation in Finland. Are there any other genetic variants with similarly high recurrence? If not, what is so unique about the Finnish variant to explain the higher recurrence?
Yeah, so I think that thank you. I think that the high rate of recurrence from the the Fin major variant in Finland is that protein is not expressed in these children. So when a transplant comes in, it has nephron in the allograft. So there may be an immune response to this new protein, which could lead to a recurrence. Right now, so it's not so it's really the response to like a protein that the body hasn't seen before. There are not other genetic variants with a similarly high recurrence. Most Mendelian forms of nephrotic syndrome do not tend to have a recurrence. It's really steroid sensitive or immunosuppressive sensitive variants. Forms of disease that do, and work is ongoing around the world to try to understand the genetics of recurrence.
Okay, thanks. Another question from Dao Fu Dai. Any data on gene interactions with environmental or lifestyle factors in the nephrotic syndrome studies?
Yeah, so even when we don't think about genetics, we recognize that there's this association of steroid-sensitive nephrotic syndrome with IgA-mediated food allergies, asthma, atopy, eczema. So I think that we're going to find over time that some of the genetic architecture of immunosuppressive sensitive nephrotic syndrome is mapping to the HLA and other immune regions. That is saying that if you have a genetic setup with your HLA or some podocyte genes, that you may end up having more susceptible. You'll get the disease, more likely to get the disease if you're exposed to some sort of allergen or infection. Environmental interactions for APOL1 associated kidney disease are very strong. What we know is HIV is a powerful second hit that potentiates APOL1. Exogenous interferon administration for hepatitis C treatment is another one. COVID-19 with interferon response cytokine storm is an environmental factor. And we found the Neptune study, and then moving forward, work by a bunch of different investigators has found that prematurity or preeclampsia among children with a high-risk APOL1 genotype may somehow interact with the kidney to ultimately increase risk of FSGS. So I think that the world of kidneyomics is really open for people who are interested in, you know, classic epidemiology and environmental science as well, because, you know, it's not just the nucleotides. We need people who are careful epidemiologists who are doing other work to really identify these second hits.
Great, thanks. There's a question in the chat from Bob Colvin. How do you decide a variant is pathogenic?
Oh, there's actually very strict rules for pathogenicity that have been laid out by the American College of Medical Genetics, ACMG. So if you Google ACMG classification system, it's a combination of metrics that include the frequency of the variant in a healthy population, the conservation of that genetic variant across life. So if an amino acid position is invariant between sea anemones and humans, it's likely to be important. So that's population genetics and comparative genomics right there. Then there's prediction algorithms about, you know, predicting the impact on the protein. And then it's very important to do, when possible, segregation with families to see if there's a healthy individual in the family harboring this variant, it's much less likely to be pathogenic. But not everyone, you know, clinical labs, CLIA certified labs need to follow these rules to attribute pathogenicity. Research groups and you know, journals and publications or things don't need to adhere to those as well. So we need to be really careful when reviewing the literature to see what kind of filtering pipelines are being used to attribute pathogenicity. And that a major work ClinGen is really to get after that concept of what is pathogenic or not.
Great, thanks. I'll just ask one more question before we close. This one's also in the chat, so you may be able to see it. You're talking about the potential role of common variants in, for example, patients with SSNS. What about so-called polygenic risk scores for glomerulopathies?
Yeah, absolutely. That would be the idea. So for the group, not everyone might know this concept of polygenic risk scores. So it's the concept that if you take a group of patients with steroid-sensitive nephrotic syndrome and compare them to those without the disease and look across their genome, the collection of SNPs or polymorphisms or variants harbored by those with disease can, through complex math, not complex, just through, you know, established methods, can converge on a score, a set of variants that, when present, increase the risk of disease. And then you can basically give weights to those variants across the genome, and then you can go to any person in the UK Biobank or your local biobank, and you can give everyone a score. And if the polygenic risk score is working, you could take a score that's built, let's say, in Neptune, or the study that we published in KI, and we could take that score and we could go to like BioView at Vanderbilt, or BioMe at Mount Sinai, or the UK Biobank, or et cetera. And then we could say, okay, let's give everyone a score. And if that score, then let's see, are there any diseases associated with that score? And what we would expect to see is that patients who have that high score are more likely to develop nephrotic syndrome or have nephrotic syndrome. So if that ends up working, it's been shown to be very effective with other diseases. And there's a lot of cool things that we could do by developing these polygenic scores and testing them in other populations. So I think that is one of the future areas of interest for us, and we are actively working on trying to figure out how to do that.
Awesome, good to hear. Okay, thanks again, Matt, for the great talk and for fielding these questions. Just a couple of quick announcements. Our next lecture in this webinar series is in 2 weeks' time, December 16th at 11 a.m. Eastern. Dr. Laura Mariani will be talking about therapeutics and the role of clinical trials in FSGS. So building on our previous discussions over the last month or two. And a recording of this webinar will be available on the RPS website within the next week or so. So thanks again, everyone, for your participation. Thanks again, Matt, for joining us today. And yeah, see you all soon.
All right, thank you.